Okay, welcome to our first pickup webinar of the fall 2022 semester. A couple of announcements before we go ahead and get started. We do have other pickup webinars planned for the rest of this fall. We don't have dates for those, but watch your email and the pickup Slack channel. They look like they're going to be good once we get them actually scheduled. The other thing I was going to mention real quick is that if anybody here is new to pickup, you can find out more about the partnership at the pickup website, which I'm going to drop a link to in the chat. And with that, I should go over the sort of standard thing. We would encourage you, if you have questions, to go ahead and either do the raise hand button or put them in the chat, just to avoid the fun Zoom trick of people talking over each other. So if you have questions, you'd like to ask something, please feel free, but use the raise hand button or put them in the chat. And then oftentimes we can, once the speaker knows there's a question, we can, actually let you talk. Okay, with that, I'll give just a real quick introduction. Our speaker today is Lisa Lapidus, which I believe I said right.
Yes, you did.
I had to ask first. Lisa is at Michigan State, where she does experimental biophysics. But the interesting thing that she's going to be talking to us about today is adapting computational physics into a intro physics class for life science majors. So with that, we'll turn it over to Lisa.
Thank you. So thanks, Nick, for the invitation. I have never spoken to this group before, and I've actually given very few education related talks before. So I hope I pitched this at the right level. What I'd like to tell you about is this entirely new curriculum that we've developed at Michigan State over the past about 5 or 6 years. And so I was going to give you an introduction to the curriculum and then show you how the computing works within it. So this is an introductory class for life science majors. And we started out several years ago trying to revise the introductory physics curriculum. And so one question you could ask is, well, why do we need to revise it? So at least at MSU, and I think at most places, life science students are usually required to take 2 terms of physics. I think it's traditionally algebra based, but we discovered in the process of doing this that our students actually have to take a certain amount of calculus. And so there's no reason we can't include some calculus in this course. But the curriculum that we offer them is really not that different than the curriculum that you would offer physics majors or engineers. And it hasn't really changed very much for the past 50 years or something like that. We start with ballistics that were developed in the 16th century. And the second semester is electrostatics that you would have developed in the 19th century. And so these are all very old topics. And then if there is, if there's time, then you generally have a grab bag of topics that are suitable for engineering, but not a lot of biology. And if it's seen at all, it's basically trees and skeletons. It's highly simplified biological topics, and they tend to be at the macro scale. And I think what a lot of physicists may not recognize is that there's been a dramatic revolution in biology over the past 50 years to understanding things more on the molecular level. And at the same time, the curriculum for introductory biology has also moved from the macro scale to the molecular level. So these students, regardless of whether they're going to study, ecology or molecular biology, have all been introduced to biology on the molecular level. And so we don't necessarily need to work at the macro scale. And there's a good reason that we should try to get away from the macro scale, except it's mathematically very hard. So here's an example that I love to show everybody. This was a paper that came out. Let's see if my video will load. Yeah, this is a paper that came out and they made a little video about it in Science, where they did these really complicated experiments with a parrot flying, and they created a sheet of lasers so that they could then watch the turbulence that was happening in the air. And the reason that everybody gets, love this video is that in order to keep the parrot safe, they had to make these 3D printed goggles for the parrot. And so that's the thing that makes it kind of fun. But nonetheless, what they were able to do once they had all this data was analyze it. And what it comes down to is a very, very complicated vector field of the motion of the wing. And this is a very difficult mathematically to understand, and is certainly well beyond the ability of an introductory student to understand. One vector is enough for them to have trouble with. So what you have to do in order to simplify this is basically take out all the things that are interesting about the biology of the wing, and you're basically left with Angry Birds, which is not very biologically relevant. So on the other hand, the physics of molecular and cellular biology is actually pretty simple, because you can use the same kinds of examples that we use at the macro scale, cannonballs and point charges. That's really what molecules are most of the time. And so we really can, without much simplification, actually talk about what molecules really do. But if you really want to understand this, you are going to have to use some calculus. And as I said, we're surprised to discover that our life science students actually do take a 1-semester survey of calculus. And so it's enough calculus for us to be able to talk about derivatives and integrals. But of course, the complicated thing about working on the molecular levels is you actually have to consider all the molecules. And that's particularly important for talking about water. You can't ignore water the way that you could air for most macroscopic examples. And so this is obviously very messy mathematically, but this is where we can bring complication in. So we developed this as a 2-semester sequence, and we organized it around 2 major themes that are usually not discussed in an introductory class. So the first semester, which of course is our traditional mechanics semester, is organized around the theme of diffusion. And we approach it as an emergent phenomenon of just a collection of elastic collisions. So I will talk in great detail about that when I show you the computational examples. And then the other semester, which is our traditional E&M, and then modern physics semester, we center that around dipoles as an arrangement of positive and negative charges, because you can discuss an enormous amount of molecular biology in terms of what has a dipole and what doesn't. And so there's quite a lot of useful information. So we use that as our primary electrostatic example. So just to sum this up, this is what our standard curriculum is for introductory physics. I don't think it's too different from what other schools use. And these are all the things that we've eliminated. And I'd be happy to talk about why we decided to eliminate each and every one of them. I have a reason for all of them during questions if you have them. But what we then substituted in were these topics, where we start the first semester with kinematics, the way that you would normally start it, and we talk about conservation laws, but then we jump very early into understanding diffusion as a series of collisions. And we keep that theme through the end of the semester, and you can see that by the end of the semester, we can actually approach statistical physics using diffusion as the example. In the second semester, we cover E&M, where we're focused primarily on dipoles. And then because we understand dipoles, we can use that to develop a model of light. And the end of the semester covers the interaction of light with biomolecules, covering things like photosynthesis. So when I decided to do this, actually, when I proposed that this was something we ought to do in this very generic way for actually promotion documents, and then my chair said, hey, you should actually do that. I was also told that MSU was pushing to transform all of our introductory classes into active learning. And so because we were starting from scratch, we decided to do that. So everything in this class is problem-based. We've also developed integrated labs. So we essentially run a studio class where lab equipment is taken out when it is appropriate within the class period. And there are absolutely no lectures other than me standing up and saying, you know, today we're talking about, well, Ohm's law. That's what I'm going to teach tomorrow. And everybody gets started on this activity. So instead they do all the work, you know, all the reading and the videos before class. And then during class, they work in groups of 3 to 6, and these are multi-step open-ended problems. And we have TAs and LAs that circulate in the classroom to assist with the work. And then we don't leave them completely alone. We do sum up what we've been working on as an activity that's typically a half an hour to an hour type of activity, where we have what we call a board meeting, because the students work on whiteboards, portable whiteboards, which they can then come up and present to the class. And so that's our way of sort of pulling together all the ideas that we wanted them to get out of the activity. And just to show you how we have progressed in developing this course, the first time we gave it in 2016, we had 30 students, and we have ramped up to almost all life science students at MSU, which our steady state we expect to be a little above 700. And so we're offering this class to most life science students now. And if you want to know what our real secret was, the thing that happened in 2021 was we got a new building, which gave us very large active learning classrooms. And that's actually what made it possible to scale up. Okay, so that's the kind of introduction to the curriculum that we have. I'm happy to answer any questions about the whole curriculum, but I was going to move on to the computational part of it. So we integrate as part of these activities. We of course do just traditional written problems, you know, a car moves down a road at a certain velocity, that sort of thing. And as I said, we also do some lab activities, but then we have a set of computational activities, and of course we use Python. But we don't require a prior course in Python because the students have absolutely no experience with this. These are life science students, and their level of computation is basically 0. And actually their computer skills are not that great either. So sometimes they have trouble with like file systems on their PCs and Macs and things like that. So we have developed some videos and some homework problems that just introduce the most simple concepts of computation, you know, what are variables, what are loops, to get them to engage with the code, because if we don't make them work with the code, there will be a subset of students that just avoid it during class. And our goal basically is to get them comfortable with seeing what code is and recognizing that they could actually understand what the code does, because the most likely computational activity they might have later on, if they are doing something in biology, would be to edit a script, because scripts are used all the time for doing all kinds of computational activities like aligning gene sequences and things like that. So we really just want them to be able to push into a code and actually see what it does more than anything else. And so we use the concept of the minimally operational program, where when we give them the blank code, it's not blank, it is a program that will run and it will do almost everything you want it to do, but some part of the code needs to be fixed, and it's usually a piece of physics. So they will actually need to edit a line that will actually get the physics correct, not necessarily a computational aspect of it, like setting up a loop or something. And of course, we use these to model complex phenomenon, or to model things that have complex analytical solutions, like a dipole field. So very much our approach is to use the activities, not just to teach them something about the physics and something about the computation, but to also basically promote some propaganda for, if you really don't like doing all the math, consider using computation to make your math easier. And I don't know how successful that is in terms of propaganda, but I think a few of them have been convinced that maybe computation is something that they should keep in mind in their future endeavors. Okay, so let me take you through how we discussed diffusion in the first semester. We can, if we want to get to diffusion, we can do this by building up from just two balls colliding. And of course, by the time we've done this, we have covered kinematics, so they understand how objects move, and they understand that momentum is conserved and energy is conserved, and then we introduce the elastic collision. So the first code that they look at, and now I'm going to see whether this actually works. I'm going to show you the codes we use, and we use GlowScript to store all our codes. So this is the code that we give them. It's complicated, so they spend some time actually telling us in words what certain lines are doing, so that they actually have to read what it is instead of just kind of having their eyes glaze over. But this is the part where it is doing the loop and basically using Euler's method to move the object. And what you can see is this is the place where the momentum is going to be transferred. But when we give it to them, the momentum is set, the change in momentum set to 0. So when you run it, you get this very non-physical result where they just pass through each other. So it's really easy for them to tell that that's not correct. So then they have to spend some time thinking about what that delta P should be. And we, prior to this activity, have already worked out the formula for that. So once they get the correct physics going, they will then have a delta P that's correct, and now they can see that it actually bounces off. And so this is kind of the nice first activity that they have where they can see, okay, I actually fixed something and now it looks right. So once they can do this in 1 dimension, then we move to 3 dimensions, and we don't have them derive the 3-dimensional momentum transfer because that's super complicated with the dot product and everything. So we just give them that code. But then when we run it, what they see is this problem. In order to run this, we always set up a little box and the balls just run out of the box. And so they have to understand what happens when the balls hit the wall and how the momentum is transferred versus the wall. So once they get the balls in the box, what they have is simply a set of if-then statements to reverse the velocity as it hits the wall if it's near the wall. And so once they do that, they can then make the balls stick around. Okay. All right, so now they've got 2 balls colliding in 3 dimensions and they have it in the box. So there's really nothing to stop them from putting lots of balls in the box. So now we really can say, okay, this is what diffusion looks like. But you can't follow them all. So you want to compute many of them together. So now we are teaching them how to set up many balls, and we can give them different properties. So this is a list of balls that have random positions, random velocities. You can also change things like the color. Later on, they will set up things like charge. They can change the mass, the size. All of these things are easy to change in this VPython. And there's 2 different collections of balls because they're supposed to represent 2 different types of molecules. In this case, we want them to think of it in terms of water molecules and then some kind of a protein. And particularly, we emphasize green fluorescent protein because that's something that is used regularly to watch molecules move around in cellular environments. And so that's the protein that they found in a jellyfish, and it, you shine light on it and it just glows. So when we run this, you can now see that there's lots of balls, but they all look the same. So you can't really tell which ones are the proteins and which ones are the waters. So then we get them to change the colors and the sizes so they can tell the difference. And so here's an example of what we think real diffusive behavior looks like. And obviously, it depends on the randomness of it. But this is basically lots and lots of water molecules and one big protein. And the protein is interacting with the walls as well, but you can see that there is some random behavior. It's being deflected by the interactions with the water molecules. And so you're getting a track that looks kind of like a random walk. But importantly, they can also go in and make the visibility of the water turned off. And then you can see the random behavior that you would see in a actual microscope experiment, where all you see is the protein apparently moving randomly. And so you can attribute it to all kinds of magical thinking, but obviously what is behind it is actual collisions with actual water molecules. So it's important for them to see that even if this is what they might have observed in an experiment, that they have to recognize that this represents many, many elastic collisions of water molecules. So I guess I can just pause there and see if there's any questions, because I just showed you a whole lot of codes.
I actually had a question. So this is showing my ignorance of biology, but can you actually do that experiment? Can you actually watch those green glowing proteins moving around under a microscope?
I forgot to put in the video that shows you that, but yes. So microscopes are really good now. They can actually detect individual molecules, and proteins can be made to glow either by forcing attaching a fluorescent molecule to them, or this particular jellyfish protein, green fluorescent protein, just glows by itself. And so you shine a laser into a microscope and you can collect the motion of molecules in the microscope. So yes, easy to see. So there's another hand from Ruja. Yes.
Yeah. Very, very nice thing. This, I really liked it. I have a quick question. So this will also be tested in the exam, or this, I mean, students also are asked test the codes or during the exam, or how is it incorporated in terms of evaluation?
So that's always been a question we have had. So we have in the past offered, put Python questions on the exam, but because we don't want them actually using a computer during the exam, we don't actually have them interact with the codes we've developed. We will give them a piece of, a very short piece of code and ask them to calculate something or tell us, what's the output going to look like, which is similar to what we do in some of our homework problems. But honestly, when it comes time to make the exam, and we have to make an exam every time, what we often end up doing is saying, well, okay, it's really much more important that they understand, momentum principle than the Python. So the Python often gets pushed aside, and the students are perfectly happy with that because so many of them really dislike it. So the appropriate thing to do would be to test them using the kinds of homework problems that we use, but it often doesn't get tested.
Thank you so much.
J West 4, you're muted.
Oh, there we go. Now I'm not, sorry. So first off, I would agree with that in terms of the testing. I would actually think that what they do in class with that would be enough to satisfy me in regards to testing or seeing what they're doing with it. But related to your follow-up comment with that, what was the student overall reaction when they heard for their intro class they're going to have to do programming? Because I'm sure that swept like a virus through the students and panic ensued immediately. How is the reaction to that bit of foreboding been?
We try not to make it sound too scary. And so what we tend to get more is a lot of grumbling. But it's very mixed. Some of them will say, I hate the Python problems. And some of them will say, oh, I never realized how exciting it is to use computing, right? So and you try to remember those guys first, right? Yeah, we used to have a problem that we taught in a room that was unair conditioned, and it seemed like every year the very first Python problem was the day it was 90 degrees. But it seems to have gotten a little better. Maybe we've just gotten better at teaching it, and so maybe we're a little less uptight about it. But we just try to keep sort of saying, look, this is one of the things you're going to do. Many other things will happen.
Well, once you have him in the class, I could see that. But again, I would be worried about there being just a potential barrier, as it were, to getting students to go ahead and come into the class. Some of them, they're already math phobic, they're already science, or at least physics phobic. Programming I think just stacks that kind of high.
So I have lots of thoughts on that, but maybe we should talk about that.
Okay, thank you.
I see one more hand, Linda.
Yes, thank you, Lisa. So this curriculum kind of reminds me of matter and interactions, which we incorporated in our calculus-based for physics majors several years ago. The feedback we've been getting from some of the biology people who have taken it and have told their friends is that it seems easier than our algebra-based physics. So I am very excited by this. I think there's potential for us to actually offer something like this to our life science people. Mm-hmm. But I'm curious about lab equipment, whether you do activities that incorporate some of the ideas and what
that's
those.
My next slide.
Oh, I'm sorry.
No, that's fine, but that's a great segue. So could I move on and then take some more questions later? Would that be okay? Okay. So this entire set of diffusion activities takes about a week for us. But as I said, we have a lab component. So the very first thing we do after this is an actual experiment. And so what you can use are polystyrene beads that are 2 microns in size. They come in lots of different sizes. They're easy to buy. And a standard microscope that has a camera. So these are not fancy microscopes at all. You can buy them for, several $100s. And so this is a set of these polystyrene beads, and they'll collect a movie like this. And so you can actually see these beads. Some of them are moving, some of them are stuck to the surface, but you can see the diffusive motion, right? So it looks a lot like what they saw before. So one of the things we do is ask them to kind of compare what this motion looks like compared to the motion they saw on the computer. And of course, the big thing is that none of these beads are moving nearly as much as they are on the computer, because the density of the balls in the computer is much lower than water density. But you can use Tracker to track one of these beads frame by frame, and can actually measure a diffusion coefficient, which they do at the end of the semester. So this is one sort of activity that we do, but this is probably the most unusual type of equipment. Most of the experiments that we do are pretty standard physics labs. We do actually have air tracks and IO labs and that kind of thing as well. Okay, so as I said, we do this very early in the semester, so we actually have not even talked about force yet. We are having them transfer momentum, and effectively what they have is a hard sphere potential when they do the collision. So once we have done that, then we go back and we do force, and we emphasize the momentum principle instead of F equals ma, because there's just not that many occasions in biology where you have a constant acceleration. And then we have to give them an atomic level force. And so what we use is something called the Leonard-Jones potential, which is basically an analytical form of the van der Waals potential, which you see down here. And so they can just program that into the computer. And so now they are looking at a code where the delta P is now actually the momentum principle, the force times the change in time times a vector that we have to put in there. And we give them a force that is this Leonard-Jones potential that I showed you before. And because this is actually a attractive potential in certain distances, they can actually see bound states. So let's see whether we can get that to work this time. So look carefully at the white balls. You can see some of them are kind of sticking together. Sometimes they start to circle around each other. And what we're showing below is I believe the kinetic energy. So yeah, you can see like right here in the middle, these are two balls that have become attracted to each other and are staying there. So that's important because we do spend a lot of time talking about bound states because that's of course important on the molecular level. And they do understand binding because every single one of these students has taken chemistry already. And so they do have some chemistry knowledge and understands what bonds are. And so even though this is simple, this is just conceptually the same as a modern molecular force field that is used all the time in computational biology. So if you know any computational biology, these are kind of the standard force fields. But this is what's done regularly by computational biologists to understand binding between proteins, protein folding. In some sense, this is also part of the structure prediction phenomenon that has taken over the field in the past couple of years, where AlphaFold can now predict every protein structure from sequence. And so we always end up showing them some kind of video that lets them see the effect of what happens when you do molecular dynamics like this. So this is the one I like because these are my collaborators, and it's also easy to understand. So this is actually a pretty huge tour de force of simulation where they simulated the entire cytoplasm of the cell with every single molecule. And I collaborate with Michael Feig at MSU, and this is currently part of my research, is understanding how do molecules interact in extremely crowded systems. So this is an artist's rendition of how they all come together, but this is what the simulation looks like. So what you're looking at are large, colorful things are the proteins. They're very close together. And then you're also looking at lots of small molecules like ATP, like glucose, things like that. What you don't see here are all the water molecules, because again, we tend to take the water out so you can see the other stuff. But what these little molecules particularly are doing, they're just diffusing the same way that they watch diffusion on the computer with the Python. So that's where we end things with force, but then of course, the end of the semester, we really want to approach statistical mechanics. This course, the traditional course usually offers a little bit of thermodynamics, but it tends to be taken from the perspective of how do you build an engine or something like that. But statistical mechanics is incredibly important in molecular biology because you are working with things that are actually countable and have statistical approaches to them. So as I mentioned, we covered diffusion really early, and then we can come back to it after we cover force. And then we can basically say, okay, you remember we did all of this stuff. So now let's just do something simple. Let's run this code for a little while. Let's see if it actually runs. Here we go. So they don't have to do anything to this code to make the force work and everything. But then at the end of the code, it gives you a snapshot. And so that's the arrows are just showing you the velocity vector for every ball, and the ball is the position. And so we can say, look, if you do this a bunch of times, you're going to get a different snapshot that's completely unique each time. But what you'll find is that the kinetic energy from all of these balls tends to be the same every time. And they can study how similar or different it is based on the number of balls that they have in there. So this is the model that we can keep referring back to of this is what a microstate actually is. And many of these microstates will have the same macro state of same kinetic energy. And entropy is simply counting up how many microstates you have. Now, this, of course, is very different than the way they have learned entropy. Entropy is this kind of magical thing that is all about disorder and, you know, concentration gradients and things like that. But we try to drill it down to being something that's very, very specific so they can actually understand what a microstate, how to construct one, and how to use it. And so then, of course, we can get to temperature instead of as this intrinsic thing that they don't really know anything about, is that, you know, it actually is the average kinetic energy. And as I said before, we of course have talked about binding, so we can then go back and explain enthalpy to them. Again, this is a concept they've seen in chemistry. And then we put it all together in the Gibbs free energy. And the Gibbs free energy is the appropriate free energy formalism that you want to use in biology, because there's no pressure change in biology, right? You're not making a Carnot engine or anything like that. And they, again, have seen this in other classes, so they have some understanding of it. But what we try to do is really try to give them an understanding that is much more physics-based. And so one example of that is the hydrophobic effect. The non-polar substance that you see here, those orange ovals, imagine that's a some kind of an oil molecule. And the blue balls are waters, and they learn that water forms hydrogen bonds with each other, so they want to interact with each other, but they can't form hydrogen bonds with the oil, so they tend to form these cages surrounding the oil. And so two oil molecules in individual cages use more water molecules than if you put them together. And so therefore combining those two oil molecules into one cage releases water into the bulk, and therefore increases the entropy, because there's low entropy in the arrangement of those waters in the cage. So the fewer waters you have in a cage and the more you have in the bulk, the higher your entropy. And of course, you can take this all the way up to actual oil separation from water, because you can just keep going. And then if we put it together with Gibbs free energy, we can actually ask, well, okay, does the enthalpy change? And it turns out that it doesn't, because whether the water is in the bulk or it's in one of these cages, it's still forming the same number of hydrogen bonds. So that really doesn't participate in any way. And so the hydrophobic effect is entirely an entropic effect. The entropy goes up as the oils come together, and the free energy then goes down. So that means it's spontaneous. So that's one way for them to use the entropy knowledge with the formalism that we use. The other way is a highly specialized way, but it's kind of cool because it uses a actual piece of research that people still use today. So this is sort of our capstone activity at the end of the semester. We want to understand the statistical mechanics of forming a piece of a protein. So the protein that we're talking about is this ribbon diagram you see up here. This is kind of the standard way that you show protein structure. It's sort of giving you the contour of the protein. And we're going to actually not look even at the whole protein. We're going to look at this last little piece, this red and yellow arrows, which form what we call a hairpin, like a hairpin that goes in your hair. And so these are just different views of that hairpin. And so the way that this model was developed, and this was actually developed in the lab where I did my postdoctoral work, so that's why I used it, is it's actually an icing model. So if you've used an icing model in magnetism and many other physics models, this is exactly the same idea. You just have a couple of states. So each amino acid is either folded or unfolded. And then you can count up the microstates that you have by assigning an enthalpy and an entropy for each amino acid in the folded state. So by this point, they actually understand what each of these words mean and how to assign them. And the code that we use is a very different kind of code, because of course there's no diffusion here. All it's doing is a bunch of bookkeeping to understand all the different microstates it can have. So this is a very different kind of code, but they do work through what it means, where they add up all of these different pieces as they count up all the microstates. And the thing that they actually have to put in is just the total delta G at the end, which is, where is it? Oh no, this is finished, right? So they have to actually calculate the delta G that they know at the end. And so if you run this program, you get this kind of interesting piece, you get this interesting set of graphs where it's showing you the free energy as you get more and more of this protein folded. And there's a barrier here, and they actually do understand what energy barriers are. They've learned about them in chemistry. And so they do understand that to go from completely unfolded to completely folded, they have to go over this energetic barrier. And at least at this temperature, the lowest energy state is here, and it is lower energy than the completely unfolded state. And then there's actually a different way to look at this, which is where is the piece of the protein that starts the folding? And so down here, you only have one amino acid in the folded structure, but it can start anywhere. And up here, you have all the amino acids in the folded structure, and the color is giving you the free energy. And so what it's basically telling you is that you want to fold this protein by going straight up the center, which is basically zip up the hairpin. You know, it starts open like this and it kind of zips up. And this is actually an important theoretical understanding of how protein folding actually works. And they have basically worked this entirely out from the statistical mechanics. So those are the kinds of examples that I was going to show you that are computationally oriented. I'm happy to talk about the rest of the curriculum if you want to know more about it. I should tell you that there is a paper I wrote about this that kind of goes through what I just said, but in more detail. And this website is a website I just created that basically has all of our activities in a pretty easy to copy format. You know, it's all in HTML, so you can basically take these activities and use them. And it is in the order in which we believe they should be used, which is why we created our own website rather than just throwing all of the activities individually onto one of the portals that like Compadre. So this is actually in the order in which we think it's appropriate to teach this class. And then I just wanted to acknowledge members of the physics department that have helped me create the class. And I'm happy to answer any more questions.
All right, we've got time for questions.
J West 4.
There I am. Okay, thank you. Quick one about just resources for this. So you have 600 students in this class this semester. We would not have anywhere near that many. And so for us to split off our life sciences from a single class would probably not be practical. But in your case, how does the staffing level, you mentioned TAs and RAs in the classroom helping with the active learning. How does that compare in terms of resources and size of the class and that to what the students were in before this?
So the reason that we actually made the studio class is we realized that that was the only way we could afford to pay for it. We already had a lab class that was separate, right? So we had a lecture class that was one number and a lab class that was another number, and all the biology majors had to take both. And they were not usually taken at the same time, and they had almost no crossover between them. So the students didn't really find the lab class all that relevant to what they were doing. So that's actually been, I think, one of the biggest selling points of this class is like you get it all done at once and it actually goes together. But what that meant was that as we pulled people out of the lab section,
you got more for the
grabbed the TAs as well. And the TAs spend more time in the classroom than they did in the lab class, but we have reduced their grading load. And the LAs was always supposed to be the thing that was going to save us, but you know, we haven't had a normal budget year since we started doing this. In 2020, you know, the budget went to hell. And so we don't actually use that many LAs. We usually only have about one per section, and our sections are 120 students.
wow.
So the
Okay.
only thing, so the main things that we had to adjust were, one, we had to have a classroom where we could put 120 students in. And so that required actually building a building where we designed the classrooms. But it has been really rewarding now that we have it. And then the other thing is that we need slightly more faculty because we used to teach lectures of 270 and now we're teaching sections of 120. And so that is basically the only increase in resources that we've had to come up with.
Thank you.
Rudra?
Very nice presentation. Thank you so much for sharing. You mentioned that all these codes are available for anyone to use in this website, right?
Yes.
Yeah, I just checked the website. I click on that, but I didn't see, like, probably.
I don't think you can click on it the way it is, but I can click on it and show it to you. We can also put this in the chat.
Oh, okay. Okay.
So let me do that. There you go.
Yep.
Yeah. So yeah, so basically this is still a work in progress. You know, there may be typos and things like that because we had all of our materials on a learning management system that everybody uses. And so I spent, you know, a month basically converting everything into a web page. But they're all web pages now, so you can just take them and use them as you want. And we have actually been working with a couple of small schools that are implementing the curriculum. And so if you guys are interested in this curriculum, I'm really excited to work with people, and I'm starting to develop a little bit of a community, still very small, where hopefully we can all help each other out.
Thank you. Thank you very much.
Nick?
So this looks fantastic. In the way our sort of algebra-based physics course enrollments work out, we sort of have three distinct populations. We have the life science majors, you know, they're biology, pre-med, that sort of thing. We have the what I'll call the engineers, which are construction management and other assorted things. And then we have a big population of kinesiology students. Do you have anything like that? Would this work more for, you know, these students that are sort of, they're not microbiology, they're sort of very, very macro human biology students?
So the way I've approached this, and kinesiology is a little bit separate. We have kinesiology as well. I think every big sports school has a kinesiology program. But the way I've approached this is that if the bio major, regardless of what length scale they are studying, and we don't have a single bio major. We have, I think, 17 majors, and they're all in different departments, and it's really confusing. They all have to take this introductory biology class, which is a pretty standard curriculum. And if you pull open almost any modern introductory biology textbook these days, it starts on the molecular level. It starts with, this is DNA, this is RNA, these are proteins, this is the Krebs cycle. It definitely starts at that molecular level. So there really isn't any reason that they can't understand it. Now, we had one school, one of the small schools in Michigan, ask us whether this was appropriate for physical therapists. And if the physical therapists really want to know about torque, then we're not teaching them about torque, but I'm not completely sure that they're really learning about torque in their physics class, right? How important it is there, I'm not completely sure. But that is something that we just have taken out because it is just not important. So we have definitely had our kinesiology majors take this class. We have not pushed really hard for kinesiology to require it because they sit outside of our normal natural science majors. And then we are still trying to decide how much we're going to keep our traditional algebra-based lecture class that has kind of the traditional curriculum for people like the construction management. And the other thing that we have a lot of is packaging. We have a packaging school that's considered to be one of the best in the country. And so there's a whole bunch of these people taking it. And so we don't know, but you put all those people together and they are a minority, you know, pretty small minority of the life science students that we serve. And I think that is probably true at many schools that, you know, even if it's like they're nursing majors or something like that, this is completely appropriate.
And just a follow-up question on that. So in the other programs that you've, I mean, I assume you had to work with other programs to get this launched. Do most of them currently give students the option to take the sort of traditional algebra based or this life science version, or have they sort of said you must take the life science version?
So far, nobody has required it, but we've been doing lots of selling. We couldn't actually take all these students until last year, right? If they had required it, we would have been really screwed because we didn't have a classroom for them. So we are now at the point where we are going to probably start asking these programs to require it. And all the feedback we're getting is that they love the curriculum. The only 10 comments we tend to get that are not completely positive are many of the students don't really like the active learning. And so since it's going to be a choice between the lecture, the traditional lecture, which is a lecture, and the active learning with the biology, they're really making two choices there. And so I don't know how that's going to go and how much pushback we're going to get. I don't think the biology advisors really are particularly sympathetic to them saying, I don't feel like doing active learning. Especially since I think a lot of what they don't like about it is that they have to actually show up to class all the time. So we'll see. It's a process. We're going to basically start asking them to require it soon. Floyd, you're muted.
Okay, so two questions. So one, what year are the biology students when they take this? Because our biology majors like to postpone physics to the last possible moment.
Yes.
And the second question is, how does this impact the pre-meds who have to take MCAT?
So that's completely the situation we have. We never have freshmen. But I think it's pretty evenly split between sophomores and juniors. And the juniors are going to take the MCAT at the end of the semester, the second semester. We've gotten some mixed messages. Basically, the MCAT was redesigned 7 or 8 years ago, and I filled out one of these inventories to say which concepts we cover, and we filled out quite a lot of it. And so we don't think that we are not preparing them for the MCAT. And the students have not told me that they have felt that they took the MCAT and were unprepared. We still cover optics, which I think is one of the things that is explicitly a question that will be asked on the MCAT that looks like a physics question, not physical concepts rolled into some kind of biology question. So we try to keep the few topics that we think are explicitly going to be asked in the MCAT there. Patricia?
Okay, thank you. So I love your presentation. And in full disclosure, Lisa gave another presentation.
We're running into each other.
Yeah, but that was very interesting. So, so I have two quick questions. The first one is, so what online readings or videos or material you provide to students before they come to class? Like, did you develop all that material, or are you using a textbook?
Yeah, so there was no textbook that worked. We went through every textbook we could find, and the problem was that none of them did it in the order we wanted to. Mm-hmm. We do offer matter and interactions as an option, and one of my colleagues actually went through and said, this is the order in which you're going to read it, but that only seems to be appropriate for the mechanics semester. The E&M semester, there's just too many things that are just not relevant. Mm-hmm. But what we actually require them to read are a set of readings that are on our learning management system. And we wrote some of them, and we stole a bunch of them from Nexus, University of Maryland. Okay. Mm-hmm. And so if you go to this website, you will see some of there are a section that's called readings. You will see readings that I wrote or my colleagues wrote that are on that website. And then the readings that we stole from Nexus, I basically just send you to the Nexus website.
Oh, okay. Okay, that's good. Yeah. And then the second question that I have is whether your colleagues in the biology department, whether they have seen any stronger preparation of the students in the upper level biology courses.
So when we have shown them the curriculum, they're like, oh, this is fantastic. I wish they would take this before they took my class. But the answer is that they often don't, because they take it so late. So I had this idea that we were going to try and develop an assessment that actually looked at how prepared they were for a typical upper level biology class. And we went through the data, figuring out what was the next class many of them took. And there was almost no single class that they took. And many of them took almost nothing after they took physics.
Okay.
So it was really unfortunate. It really would be nice if we could move this to the freshman year, and then there would be a lot more that we could work on. But we can't get that changed.
Yeah. Yeah, thank you.
Okay, well, maybe that's a good place to stop. So let's thank Lisa one more time. It was a great talk.
Thank you.
And I'll turn off the recording and we will see you next time, hopefully.